Welcome, everyone. It's a pleasure to start this new cycle of seminars here at the Josep Carreras Leukemia Research Institute. The idea of this cycle of seminars, the showcasing discoveries at the Josep Carreras Research Institute, open lectures of its group leaders, is to show the world the things that we're doing here in our institute and the excellent research developed by our group leaders here. And you will see a mix of the people with an expertise from molecular biology, cell biology, to more context-related, disease-related, leukemia, lymphoma, etc. And I would like to thank Dr. MARCUS BUSCHBECK for being brave enough to start this cycle of seminars. And Marcus, he trained in molecular cancer research and chromatin biology at different institutions, such as very prestigious, like the Max Planck Institute of Biochemistry in Germany, the University of Oxford in UK, and the Center of Genomic Regulation in Barcelona, here in Catalonia. In 2009, he joined in the campus of Can Ruti in Badalona, where we are now. And in 2015, he joined the Josep Carreras Leukemia Research Institute. So he has been a group leader with us in the last 5 years. In this new role, he has worked very well the fusion between this basic and translational biology. And his research is in the understanding of the hematopoietic stem cell defects that can lead to Myelodysplastic syndrome and other blood disorders. So it's a great pleasure to have him here. And just to remind you that you can ask questions using the question mark button there in the web. And more than happy now to start to hear about science from Marcus. Marcus?
Well, thanks a lot, Manel. Thanks to all those out there in silent mode who are listening. It will be a great pleasure to start this series and to show you a little bit what we are doing in my lab, what we have been doing in the past, and what we want to do in the future. I titled my talk Studying Chromatin as Nexus Between Basic and Applied Research. But everything basically can go back to this very, very simple slide, where we basically ask, how can it be that the genetic information in form of 2 meters of DNA, first of all, it fits in this tiny space of a cell, and inside of the cell and the nucleus, which is below 20 micrometers. And the way it fits might actually decide how we use the genetic information. And this can give rise to hundreds of different cell types, which on top can be in thousands of different cell states. And this is what we want to understand, and to understand the significance of this regulation of chromatin structure for cell behavior in health and disease. Of course, we know that DNA, let's see, that should move, that does not. We know that DNA, of course, is part of chromatin. I have to apologize here. And so, yeah, so we know that DNA, of course, is part of chromatin, and chromatin structure is going on many different levels. And one of these levels on the first dimension is, of course, the packaging of DNA on histones and nucleosomes. And then we have different levels of higher order structures. We are just starting to understand. We have this medium-sized aggregates, which then go into larger-sized aggregates. In the end, this defines the territory occupied by a certain chromosome in the interface nucleus. So in my lab, we basically take 2 different takes on how we deal with this question. On one hand, we do very basic research where we ask what actually happens if we modify the nucleosome. On the other hand, we also take very translational questions where we actually ask, can we exploit chromatin regulation to improve the way we treat disease? And the diseases we're interested in, like Manel already mentioned, are stem cell defects called Myelodysplastic syndromes. Very related to that, acute myeloid leukemia and B-cell lymphomas. And the way we approach this is 80% experimental and 20% computational. So what are we going to do in this talk, which I have roughly divided in 2 halves? In the first half, I will give you a little bit an idea of the research lines we have started here at the Carreras Institute. And most of it, what I'm going to show you, will be work in progress. And in the second half, I will actually take you deep into the basic research, how we do study the nucleosome and the findings we have made in the past and the things we want to study in the future. So the very first project which I would like to present to you is the response project. The response project. And the response project is basically carried out by a few people in the lab, Janine Diesch, Raquel Casquero, who just has joined the Pablo Menendez lab a few days ago, and Michael Mayer. And here we have quite a number of important collaborators, and I would like to highlight Johannes Zuber, and you will see in a while why. So in this project, we focus on the myeloid system, and the myeloid system, you can actually very much simplify it if you draw it like this. So we go from hematopoietic stem cells, which progressively reduce their or increase their commitment and reduce the capacity to differentiate. But the important thing is that in the end, it provides all the different myeloid cell types we have in our blood. And what we now have in the clinic is very often people, old people frequently coming in because the doctors have observed reduced blood numbers. And so they come to the clinic. Here we actually analyze the bone marrow. And if we observe that the bone marrow is not normal, that we actually find anormal formed cells, weird cells, we talk about dysplasia. And the combination of cytopenia, so reduced cell numbers, and dysplasia in the bone marrow is what we, as a group of diseases, call Myelodysplastic syndrome. And apart from the symptoms these patients already have, this is very relevant because 1 in 3 of them will actually progress and come back in a year or 2 with a full-blown acute myeloid leukemia. So this myeloid leukemia basically means that we have, in addition to the phenotypes of MDS, an increased number of blasts in the bone marrow and then also in the blood. So the thing we do when we have our high-risk MDS patients, so we of course give them the best treatment we have. And all those patients which we cannot transplant, the best treatment right now we have in the clinic is azacitidine, Vidaza, or decitabine. The issue with this treatment is, first of all, it takes us 6 months to know if patients respond or not. And then we have almost half of the patients not responding at all, and half of the patients responding, but only very few of those respond completely. And the issue is that almost all responders eventually relapse. So we took a closer look at azacitidine mechanism. And what actually azacitidine is a nucleotide analog. That means it goes through a set of enzymatic reactions and in the end ends up in DNA. And the same is true for decitabine. And aza, in addition to that, also ends up in RNA. So the drug action, as far as we understand it right now, once it is incorporated in DNA, it actually cross-links covalently DNMTs to DNA. And this has 2 consequences. One consequence is DNA damage. And this needs to be repaired. Otherwise, the cells go into cytostasis or into cytotoxicity and die. And the other consequence of this is, of course, the inhibition of the enzymes, because DNMTs are the enzymes responsible for DNA methylation. So what we observe is a net demethylation of DNA, and this can lead to the reactivation or activation of genes, or in some cases, endogenous retroviruses. And this actually can elicit an intracellular EF interferon response and signal to the immune system. So we argued, and this is our hypothesis, if aza acts in the context of chromatin, because that's where the DNA is, we hypothesize that maybe by mining the chromatin regulatory space, we would be able to identify combinatorial drug targets. So how do we do that? So first of all, we got together with people like Johannes Zuber in the IMP, who became a very important collaborator, and we developed a chromatin loss of function library. What do we do for that? First of all, we use one of the vectors he already had designed in the lab, which is basically a very interesting vector because it combines a Pol2-driven promoter, a GFP transgene, and in the UTR of this GFP, we actually put in a microRNA embedded hairpin. And this hairpin then is very efficient and optimized to inhibit the target gene. In addition to that, we have added a UCO, which is a ubiquitous chromatin opening element, which should avoid the silencing once this insert has been introduced into cells. And this together makes it a very potent tool to actually achieve very good knockdown efficiencies with single copy integrations. So then what Johannes has done with his team, they have cloned into this vector hairpins targeting 912 chromatin regulators. And for each chromatin regulator, they have taken 8 hairpins. So making this a quite comprehensive library. To choose a model system, remember we want to work on MDS. We needed to get a little bit creative here because there is no MDS cell line. MDS is actually characterized by the lack of growth. So what we actually did is we did second best. We got into contact with the DSM-Z, and in there with the head of the cell culture collection, which was Professor Hans Drexler, and we asked them to send us all acute myeloid leukemia cell lines, which have been isolated from patients who before had either an MPS or a similar disease. What we then did, we analyzed the growth behavior, and in collaboration with the diagnostics unit of our hospital and also Kiko Soleil, we basically characterized all these cells as if they would be patients. And in the end, we selected 4 cell lines which really work well in our hands and are suitable model systems for secondary AML derived from MDS. Now, our main workhorse are these cells, which are poorly known, but they are called SKK1 cells. All this data has been published as a small resource paper and is available to whoever would be interested. So what we now do is we introduce our library into these cells. We do this with low multiplicity of infection to have single copy integration. We select infected cells, and now we split them, and we either keep them untreated or we simulate azacitidine treatment. And we do this by continuously adding azacitidine over 2 weeks. And actually what happens if you look at viability of these treated cells, you see they start quite well off. After a few days, you see viability decreases, but they start to recover until the end of the treatment, suggesting that now those cells who have some growth advantage start dominating, while those cells which are weaker in these conditions will start to disappear. And now we can actually use these hairpins as barcodes, and we can actually sequence these populations and count barcodes, and thereby determine which hairpins have been disappeared, which clones have decreased, which clones have expanded. And if you now collapse these 8 shRNAs per gene, we get actually this gene point of view, and you actually see here I plot AZA versus untreated. So on this side, you see those clones and those hairpins and genes which are enriched. And this includes, for example, NAA15. And this is a gene already known to link a DNA damage response to apoptosis. So it makes perfectly sense that knocking down this gene, cells will survive better under these conditions. But of course, these genes, you see, they're very abundant. They're not really interesting in the sense that we are actually looking for a potential drug target. And inhibiting these genes actually makes the cells more resistant. So we need to look on the other side. And you can see up here, there's actually one point which looks very interesting, has a very good p-value, a very good fold change. And I have been asked by my co-workers not to name the gene as of today. So let's call it chromatin regulator X for now, okay? We will resolve this issue soon. And we had to validate these findings, of course. We did this now by making stable cell lines with very unique hairpins, which we know work well. For example, here now for this NAA15, which uses a control, you see in this AZA response curve, these are the control cells, and you see these knockdown cells are clearly more resistant. And if you now use the chromatin regulator X, the 2 knockdowns really make the cells hypersensitive in respect to the control. So fortunately, others have done already us the favor to develop small compound inhibitors for chromatin regulator X. So we got our hands on them. And as you can see here, if you now use 2 different concentrations of this chromatin X inhibitor, you see in both cases, the curves of is clearly below the control of AZA alone. And if we actually calculate the combination index, we really see synergy in these concentrations. So I mentioned before that azacitidine has a very similar parallel drug, which is decitabine. Both of them are in the clinic, and there's no real selection criteria besides the amount of deals the pharma companies do with the country's national health systems. So we actually said, okay, let's have a look at decitabine. And there was a huge surprise to see that in the combination with decitabine, there's no synergy at all. And just the opposite, we see a very clear antagonism. And this is, as I said, is very much work in progress. But basically, it already lets us conclude a few very important things. First of all, cell intrinsic response to azacitidine can indeed be modulated by the combinatorial inhibition of a chromatin regulator. The synergies we find by these methods, they might actually be very specific for AZA or most likely also for decitabine. And now if you think about, there's a major difference. Also these drugs that are really used in the clinic as would be a similar drugs. AZA is doing something additional, and this is actually going into RNA. And I think we need to understand, now thanks to this screening method, we need to understand why some interferences synergize with AZA but not with decitabine. And if we understand this, we might actually even be able to extract biomarkers which might help us to go into treatment choice for these 2 drugs. So with all this, I would like to zoom back to my initial scheme about indicated cancer patients and the current best treatment, because this is actually very true for all kind of cancer patients that cannot be operated. We will always give the best treatment, and they will always split into two groups which either respond or do not respond. And in many cases, responders will eventually relapse. So the need for biomarkers and combinatorial drug targets is actually much more general. So we did basically, we got together a mini consortium that we actually call a response. I'm coordinating this, where we actually analyze 3 different cancers: lung cancer in combination with chemotherapy, colorectal cancer with a different type of chemotherapy, and what I showed you before on MDS and azacitidine. This project has been funded, is still running until 2021, and by our National Health Institute. The people who run this, we are of course not experts in solid cancer, so we have experts of solid cancers on board. We have the lung cancer group, which is Jose. We have the colorectal cancer groups of Martínez Balibrea and Sonia Forcales. Luis Zamora is working together with us on the MDS AML side, and Lorenzo Pasquale is providing expertise on mechanistic studies. So this is one of the projects I wanted to show you. Now I will give you some glimpse of a few others we are doing. One is, for example, this one. We asked the question, in addition to treating the cancer, can we also treat the bone marrow microenvironment? And this is actually a very valid question if you think about MDS, because MDS starts as a clonal disease with very few mutated clones, and they grow terribly if you take them into culture. But eventually these clones take over and dominate over healthy hematopoietic stem cells. And now what has been shown is that actually the microenvironment, and here we have drawn a mesenchymal stromal cell, what they actually start doing, they start supporting malignant cells and at the same time repress healthy cells. So we could think, if this switch has happened here, we might be able to define or identify epigenetic drugs who can revert this, and by making now an environment which favors healthy cells and not disease cells. So this is actually very much work in progress. I'm not going to show any data here. This is a grant which has been funded by the German José Carreras Leukemia Stiftung. We do this together with Katharina Götze from the TU Med and in collaboration with Hind Medjov. And the person in the lab who just has taken up this project again is René Winkler. Another question we asked is, can we treat before symptoms appear? And this goes to a novel concept called disease interception. And this is actually not a concept I have invented, but this is also a concept which has been driving the Lifetime Consortium. And they actually have very nicely discussed this concept in the very recent Nature white paper. So what is this? If we now assume that this line are healthy cells, there must be a moment when the first healthy cells become altered and deviate from a normal trajectory. And this probably happens when we have only few cells long before a patient has symptoms. So if we would be able to find these first altered cells and actually to target them, then we might be able to intercept long before the real disease happens. Because if we wait, we'll have more cells, the patient will have symptoms. That's usually the moment when we actually have patients. And the therapy will be difficult, expensive, invasive, and sometimes directly impossible. And then, of course, now the myeloid system actually provides us a paradigm for a clonal disease. Because in a healthy system, we have something like 100,000 hematopoietic stem cells at the beginning at birth to actually make our blood. And then mutations will just happen and accumulate over life. And eventually, first mutation will appear, but they will be neutral. When these mutations confer a certain growth advantage, clones will expand, but we will be healthy, no symptoms. We call this clonal hematopoiesis with indeterminate potential. And only if these disease cells now really acquire additional mutations, then we can have an MDS and at a later stage an AML. So I think this is a very nice is of course the wrong word for a disease spectrum, but this disease actually can serve us as a paradigm to study whether disease cell interception is possible. And the good news is we have received funding for a Marie Curie training network called INTERCEPT-MDS, which again I will be coordinating. We have lots of scientific partners, both combining clinic and industry, basic research and mouse model people. And the important news is that beginning of next year, we will actually start the call and be able to recruit 12 PhD students who will actually try to develop the tools and the knowledge to test the concept of disease cell interception in myeloid diseases. So here we have reached this turning point. I think I have given you a little bit of an idea of what we try to do here at the Carreras Institute. And now I would like to show you the basic research we have been doing over the last years. And this research line actually goes back a few years. I have started that as a postdoc 12, 13 years ago, and it's still ongoing. So here we ask, now what happens if the unit of chromatin is altered? And we are not interested in any modification of the nucleosome, but we are particularly fascinated by the possibility of modifying the nucleosome with histone variants. So here I've drawn together with Sandra Hake a cartoon of a nucleosome. Also, it's wrongly drawn. It makes the important point that they actually have 4 different types of histones in there, and they come in pairs. And for 2 types of these histones, the histones H2A and histones H3, we actually have lots of different proteins which are different, and they can actually replace the normal, or if you want, replication-coupled histones, and thereby give chromatin very specific biochemical properties. And you see here, for example, this very large group of H2A histones. And if you look in here, you actually see that 3 of them actually stick out because they're much larger than normal histones. And these are the macro H2A histones, and these have been our center of study for many years. So now I would like to show you one nucleosome, and now here you see macro H2A nucleosome. So you see this very beautiful nucleosome structure as Carol Luger has been describing it many years ago. If you now replace the yellow one, which is H2A, with a macro H2A variant, nothing really happens overall. There are tiny changes in structure, but they're minimal. The major changes it actually introduces, it adds 2 domains. Here, a very long unstructured linker, which actually comes out right next to the diet axis where the DNA goes in and out, and a macrodomain, which is a globular fold here on the top. And this, of course, is a drastic, a major change of a nucleosome. And to make our life a little bit more difficult, there are actually 2 genes and 1 event of alternative splicing, giving rise to 3 different macro H2A proteins, which overall have this structure. And we already know that approximately 1 to 2% of all nucleosomes in our genome in most differentiated cells actually do look like this. So what we did, we and many others, we did the biology approach. The first thing you do when you have a new gene, you remove it and ask, do we see phenotypes? And we and others have done this in differentiation development, cancer, and in reprogramming. And I would like to show you 2 quick examples. So please focus on the zebrafish. So this is an experiment I did as a postdoc. And what you actually can see, if you remove macro H2A from zebrafish in development, you get this. So what's that? So that's still a fish, obviously. We look very careful. This fish is complete. But if we zoom in, we see that development has not been robust. So every tissue has defects. The brain has defects. The eye is too small. The body curvature is very obvious. So development is not perfect, telling us that macro H2A makes development robust. In terms of cancer, we came to the conclusion, we and others, that macro H2A is mainly a tumor suppressor. And the most striking example is this one by the Bernstein lab. So what they actually have done is they've injected melanoma cells into mice. And then they take out the lung. And this lung is perfect. There's not a single metastasis in this lung. But now, if we repeat the experiment, inactivated macro H2A in these melanoma cells, the lung gets completely filled with metastasis. And this was the first and the most striking example of how macro H2A acts as a tumor suppressor. In reprogramming, I only cite 1 paper from Vincent Pask here, but several labs have shown at the same time and simultaneously that if you remove macro H2A, you accelerate or increase the efficiency of reprogramming to induce pluripotency cells. Altogether, now the very vague biological conclusion from this is that somehow macro H2A seems to help to stabilize differentiated states. So what I will now do in the rest of the talk, I will give you 2 stories about macro H2A, and actually I will link them to the 2 different domains of macro H2A. I will actually try to convince you that the linker, the one which I actually draw only with a dotted line because we don't have a structure, is actually important for regulating 3D chromatin architecture. And I would like to show you that the macrodomain has a very interesting function because it's able to bind metabolites. So all this started with a very general question, which we actually have posed at the very beginning of my lab. How does macro HGA function in chromatin? A simple question, a complicated answer. The people working on this are, for example, two postdocs, David Corujo and Roberto Malimberli, working mainly experimentally, working 100% computationally, two PhD students, Marguerite Marie Le Paneret and Olli Meus. And here a whole list of collaborators has supported us in the past. I would like to mention Andreas Ladurner, who has become a very repetitive and very important collaborator for us. And I would like to mention José Luis Gómez Gameta, who unfortunately has left us last week, and he has been supportive from the very beginning. Thanks a lot, old buddy. With all this, I would like to show you the first thing we need to consider. The first question we need to consider was, how much macro HGA do we actually have, and how does it distribute? So I mentioned before, when you do mass spec and you actually plotted how much macro HGA have in the entire H2A pool, you actually get to the conclusion it's only 1%. So even if you put only 1 into a nucleosome, that ends at 1–2% of nucleosomes. Now, if you do ChIP-seq, you actually get a different story. What you get, actually, here's the input lane. Here's just one example, doesn't really matter. And you see this very mild, spread out enrichment of macro HGA. If you now run algorithms on that and ask how much macro HGA do we have in regions enriched over background, it gives you back that up to 30% of the genome have some enrichment of macro HGA. If you put this into context with this 1% of the H2A pool, it tells us it's not very positioned. Most of the signals we see are not saturated. And now this is a very important question, or very important information when we try to start thinking about mechanism. Because obviously it must be mechanism that be more general and very difficult to nail to very specific positions. Okay, so what did we do? We actually did the zoom out view. This is an experiment actually done by former lab member Juliette Oe. But he actually said, okay, let's take a look from a distance. And this is the most distant look you can take at heterochromatin or at chromatin. This is actually a cell nucleus. It's in HepG2 cells. So these are hepatoblastoma cells from liver. And what you actually can see is this very nice round structure. You see dark gray and light gray. The dark gray is more dense chromatin, which we call heterochromatin. The light gray is euchromatin, more open structured chromatin. And this is what you actually observe. What we did, or what Juliette did, he removed all macro HGA from these cells by using very efficient knockdowns. And then he repeated the electron microscopy. And that's what he observed. He observed very, very drastic changes. So first of all, you see the nucleus is not round anymore. It actually has these deformations. And you see that everything which has been nicely structured at dark, heterochromatin and lighter stuff, everything gets into an intermediate stage, medium gray. Actually, I call these cells double knockdown cells, DKD. So this is a word I will use during the talk. If you now zoom in, for example, if you zoom in at the periphery, you see this very nice heterochromatin, dense heterochromatin in control cells. This is completely gone in double knockdown cells. If you zoom in at the nucleolus is very nicely structured in control cells. You even have this thin layer of heterochromatin on top. All this structure and the heterochromatin is completely gone in the double knockdown cells. So a different way of looking at heterochromatin is actually by staining it with H3K9 trimethylation. And then what you get in control cells, you get this puncturing aggregate staining. You see we have a few nice thicker aggregates. If you look in the double knockdown cells, this seems to fall apart into smaller aggregates. Now actually we can quantify this in an automated way. We can ask the computer to count the foci with intensity above background and to number them. And then basically we have something in our hands which allows us to describe the phenotype in an automated way. So we see more foci. Another thing we observed, and I put here two pictures which actually exaggerate this, we see an increase in nuclear size. So if you measure nuclear size, also this is significantly increased. And this might actually be a very general lesson which we probably just start to understand, that chromatin compaction is of course limited by the nuclear space, but at the same time also nuclear size might directly be correlating with how chromatin is compacted or not. I think there's still to learn here. So now heterochromatin in general, stained with H3K9 methylation, we can now zoom in specific heterochromatin regions. For example, we can stain with fish, a repeat that we know is part of heterochromatin. So this is a satellite 2 repeat. And you see it's very nicely compact in control cells. It's inside here in the nucleus. You see the dark counter stain. And this actually gets completely open and partially dissolved in double knockdown cells. And now I will summarize a lot of work in just a few lines. Because of course the question we had asked, what kind of decompaction are we actually looking at? And we came to the conclusion that there's actually nothing happening in the first dimension. So if you think about chromatin fibers and this compaction in this first dimension, this is not affected at all. The evidence we have, there was no change in K9 trimethylation on these loci. There was no change in nucleosome occupancy. There was no change in chromatin accessibility. So we think what actually happens is something interfiber, which is actually not been observed with these techniques. And when we actually look at these repeats, which anyway, they're almost not expressed at all, if at all, also we have this very drastic change. We only had a very subtle increase in transcription, which still is neglectably low. So now we said, okay, maybe we should go back to the lamina and the periphery, because remember, at the periphery, we saw this phenotype are actually most drastic. And here we see the heterochromatin, and here it's gone. And we know that the periphery is actually a very interesting area of the nucleus, because it's, first of all, when you have the double membrane, you have a lot of anchoring proteins, which now anchor a meshwork of lamin proteins to the plasma, to the nuclear membrane from the inside. And this actually contains lots of small and low affinity binding sites, which actually can interact with chromatin. And it was also interesting to see that macro-HJ was actually co-purified by others with a lamin B1 complex. So we asked, does macro-HJ contribute to the tethering of heterochromatin to lamina? We used two different experiments to do that. One was actually proximity ligation assay. Not sure if you're familiar with this, but basically what you do, you take two antibodies against two parts you're interested in our case, lamin B1 for the lamina and H3K9 trimethylation with heterochromatin. And if the two antibodies are in close proximity, this method allows to convert this into a signal. And we actually see now these signals lighting up exactly where we expect them to be, at the periphery of the nucleus. And the double knockdown cells, these signals are clearly reduced. A different way of looking at it is, of course, by chip. So we measured lamin B1 on repeats that we know are actually sitting in heterochromatin. We have a certain background. You get a signal over a background if you use the lamin B1 antibody in control cells, and in the double knockdown cells, this signal is entirely gone. So this basically tells us that what macro-HJ actually does, it is helping. Also, it's in the smallest subunit of chromatin. It helps to organize heterochromatin on a larger scale. So we can say that in normal cells, we have this very nicely compacted heterochromatin attached to the lamina, and in control cells, it's actually decompacted and has lost its interaction with lamina. The issue is we were unable to tell cause and consequence. It's possible that macro-HJ provides a binding interface to lamins and thereby helps to actually collect heterochromatin and compact it indirectly at the lamina. But it could also be that it actually helps to do the compaction itself and thereby increases the density of low affinity binding sites, and like Velcro helps heterochromatin to go back to the lamina. So, but one thing we could do is to ask which domain is responsible for that. And for this, actually, David went back to this heterochromatin foci assay, and now I show you exactly the same pictures I've shown you before and the same quantification I've shown you before. So we see more foci in the double knockdown cells. What we now have done, we have introduced YFP and YFP take macro-HJ, and what you actually can see that YFP itself is neutral, and YFP macro-HJ actually can rescue the phenotype. If we now use a mutant lacking the macrodomain, it still rescues. And only if you use a mutant which also lacks the linker, this rescue is completely lost and the mutant is neutral. So this actually told us, wow, it's the linker does the job. And that now seems logical, but back then this was a surprise because we and many others, since we didn't have the structure, most of our attention was focused on the macrodomain, and the linker was very often just cut in experiments because we just tested the two different parts. And now we know it's actually the linker that's important. We could corroborate these findings with Andreas Ladona using different assays. All this is published. So interestingly, others, and this again includes Carolyn Luger and Craig Baumann, have actually shown in vitro that the linker is important. Carolyn actually has made a very interesting find that if you add only the linker, and it only works if you only add the linker, if you add it to normal H2A, you can aggregate chromatin fibers in vitro. And Craig actually pointed out that the linker is actually right next to the DNA. So this here, right? Here is the linker, here is the DNA coming in and out. And of course, this can stabilize. And this was very nicely shown in this study. And now, of course, this puts us in a position to go back to this many initial observed phenotypes in cancer, in development, in differentiation, and reprogramming. And we can ask, is the linker responsible for that? And can we actually relate these very general phenotypes to the capacity of the linker to organize 3D chromatin architecture? Okay, now the last 10 minutes, I would like to change gears and take all your attention to the macrodomain. So the macrodomain, as I mentioned before, is a globular domain. Because of 2 genes and 1 splicing event, it comes in 3 different flavors. Here I've cut them open, and what you actually can see that they have cavities. You can see the cavities are different, and we know at least for 1 cavity, and that's of macro H2A 1.1, that it can actually bind a metabolite, and this metabolite is ADP ribose. And we know the other 2 do not. So these 2 ladies, who were PhD students in my lab, Melania Pusovic-Marjanovic and Sarah Hurtado-Barges, both of them are doctors now and have left, have asked the question, so what is the consequence of having an ADP ribose binding site on chromatin? Again, many collaborators have helped us. I would like to highlight Entropos Pisalik and Raffaele Teperino, who were really, really essential for us. And the first finding we had is what it does, it inhibits PARP1 because macro H2A binds ADP ribose, which is here illustrated as a star, but not only when it's free ADP ribose, but also as a polymer, and also when it is a post-translational modification on proteins. And one of the proteins that very often auto-modifies is the enzyme itself. And what in case you have more, and this is very important, you have more macro H2A 1.1 in cells than actually PARP, this complex is actually an inhibitory complex. You can actually see that in vitro. Here we measure PARP activity with autopyrylation, and if you titrate in a macrodomain, you can completely reduce this enzymatic activity. If you titrate in a different macrodomain, like for macro H2A2, this is completely neutral. So what is the consequence of inhibiting PARP? And the first thing is completely trivial. If you inhibit PARP, you actually inhibit PARP-dependent processes. And we have actually shown this in the context of chromatin expansion, which is important for DNA repair. But this is something I do not want to talk about. I want to talk about something else. Something which is a little bit less, people are less aware of, is that actually PARP can also be considered a metabolic enzyme, because what it does, it uses the all available nuclear NAD+, it breaks it into nicotinamide, NAM, and this ADP ribose units. And this means that if we have hyperactivated PARP1, maybe downstream of DNA damage or stress response, it can deplete nuclear NAD+. And this has actually consequences for the total cellular metabolism, because NAD+ is terribly important, and the cells will very quickly recycle it. There's very little de novo synthesis, and most of the recycling is happening. So using the nicotinamide and in the cytosol, returning it into a precursor called NMN, and then the NMN gets recycled in NAD+ by enzymes. And thereby this cycle is going on, but also the same cycle, the same central reaction also feeds the NAD+ pools of all other cellular compartments, including mitochondria. And now we'll summarize what we have found in a cartoon, and later I will give you pieces of data showing you what we have seen. So we have found that in some cells where macroHTA 1.1 becomes more abundant as PARP1, this inhibition leads to dampening the nuclear NAD+ consumption. And this actually has consequences because now the precursor can be used by other organelles and the NAD+ consumption in other organelles can go up. And this, for example, can be read out by seeing that mitochondria have improved and increased respiration and can do increased ATP production. So now the data. So what are these cells where macroHTA 1.1 is higher than PARP1? These are actually muscle cells. And you can actually nicely see that if you take a cell culture model for muscle cells, for example, C2C12 myoblasts, which are very fast proliferating cells, but only in 4 days with differentiation, you can convert them into multinucleated myotubes. And here I show you now the two different transcripts for the two isoforms. This is the one binding ADP ribose, this is the one not binding ADP ribose. We start with the one not binding, and within a day, the other transcript comes up. And after only 2, 3, 4 days, you have only the 1.1 transcript left on the RNA level. And we have measured the proteins, both macroHTA 1.1 and PARP. We have counted molecules. What we have in this situation is 1.2 times more macroHTA 1.1 than PARP1. So inhibitory conditions. So now we did the experiment. We knocked down macroHTA 1.1, and we have analyzed what this actually means for mitochondria and mitochondria. So first, what we did was measuring metabolite levels with mass spec. Here you see that we have a reduction of both the precursor NMN and NAD+ itself in mitochondria. We used C-host to measure mitochondrial activity, so respiration. This is the normal level of respiration, and again, this goes drastically down in knockdown conditions. And if I was right with my initial cartoon, I argue, basically, maybe that was not strong enough, I argued that this is metabolite-mediated communication and not going through gene regulation. And so if this is true, I should be able to rescue this just by giving mitochondria an alternative NAD+ source. And this is actually the case. If you just feed the precursor before measuring the experiment, we can fully rescue this phenotype. So what we do have here, I think, is one example, maybe one of the best examples so far, but certainly not the only example, for how distant organelles, and here I would consider mitochondria as the energy-producing organelle and the nucleus as the one which has the genetic information, that they actually be able to communicate through metabolites. And this is very nicely shown for NAD+ now. And it's interesting now to see that differences in chromatin can modulate the communication between organelles. And I think this is a very nice example. Very good. Again, if you're interested to read more about this, we have summarized this in a discussion paper. Now, the take-home message of this talk is the following. If you want to remember one thing, please remember this. We are working on the most interesting protein in the nucleus, at least what we think. We think it's an histone variant. We think it's actually macroHTA. It contains 3 different domains, the histone fold, a linker domain, and the macrodomain. The linker is important for 3D chromatin architecture. The macrodomain is actually linking this protein through metabolite binding to metabolism. So we have a protein here, which is a structural component of chromatin, and it's an intersection between chromatin structure and metabolism. So what are we doing right now? And now I will give you 2 or 3 quick snippets of what's going on in the lab. On one hand, Iva Guberovic and Sara Hurtado-Vargés have asked, how old is this metabolic function of macroHTA? So they have done evolutionary analysis, and just very quick as a spoiler. As metasome life, so it actually appeared right before multicellular organisms came. So we think it might be a first indication of differentiation. And the other important thing is the oldest of all macroHTA is actually similar to the one able to bind metabolites. So this is coming up. Please look out for our paper. If you're a reviewer, please review it kindly. The other thing we would like to do, and this is work driven by computational expert Roberto Malinverni. We would like to gain resolution on what this phenotype on nuclear architecture actually means by moving from microscopy into chromosome conformation capture techniques using genome-wide, antibody, protein focused, or locus focused techniques. And we can observe first differences, and we are still trying to understand this. Again, this is ongoing. Another thing we want to ask is
Sí,
no
jo tampoc puc veure, la veritat.
And so one way of how we address the cancer relevance is actually now going through gene regulation. So here's one example from David in collaboration with Carolina Armengol from the neighboring institute, where they have crossed macro-HTA dependent genes and cancer genes, and have asked, are there genes which are macro-HTA dependent and upregulated in cancers which overlap? One good example you see here is, for example, DKK1. Now we can take these genes, move to the locus, use Umi4C to actually ask, does macro-HTA presence and absence change the locus? And without going into much detail, the answer is yes. And with all this, I just mentioned that, of course, we are moving a little bit away from hepatoblastoma back into blood. We have funding to study this in the context of MDS and secondary AML with Katharina Götze. We have just received funding from the Marrow Foundation to study this in AML together with Florian Heidel. And we have initiated collaboration with Gail Roux, where we want to move into German center B-cell lymphoma. And if I have intrigued you a little bit concerning histone variants, then please let me point out that I have the pleasure to co-organize an EMBO workshop with Emily Bernstein. This will happen here at the Carreras Institute. So if you're curious to visit us, please scribble down these dates. Of course, we need to see how, if the health situation will allow this. Right now, we are still optimistic, and it would be certainly a great pleasure to host you here in Badalona. And with all this, I would like to thank all these people for the very great work they have been doing, for a great atmosphere, for being a fantastic group, and for making me such a proud PI. Thanks a lot, guys. Of course, I have to thank the European Commission and the foundations, and of course, the Spanish state for the continuous funding over the last years, especially the European Commission for the 2 ITNs. Chromi has finished. INTERCEPT-MDS is just to take off. And if you have more questions, here's my contact. Happy to answer any questions you might have. Thanks a lot.
Thank you, Marcus, for this great presentation. So here we have some questions for the speakers. The first question is, could you speculate on the role of macro H2A in DNA damage?
I mean, there's no need to speculate on macro H2A in DNA damage because there are several very good labs in the world working on this. Probably one lab is Philipp Oberdörfer, who has actually shown that macro H2A has an important role by deciding which choice of DNA repair is taken. So macro H2A promotes homologous recombination. There are 3 consecutive molecular cell papers showing that. There is other work coming up from collaborators of us who provide additional detail in that direction. So yeah, if you're interested in that, please take a look at these.
Thank you. Another question. Can you give some details about the experimental approach you're going to use in the interception project?
Yeah, again, if you look a little bit at the white paper, the Lifetime Consortium has just published 2 weeks ago in Nature, it will be very obvious what we want to do. Okay, interception means 2 things. First of all, you need to identify disease cells before symptoms. So initially in this project, we will of course use model systems like model organisms, co-culture systems, and the way we think we can detect these cells is with single-cell RNA-seq, because they should already have an altered transcription pattern. And with single-cell RNA-seq, we might be able to catch 3, 4, 5 within 10,000s of normal cells. And the way we want to intercept, now being a chromatin regulator guy, of course, I speculate again that the chromatin regulatory landscape will provide us the targets we actually need.
Thank you. How conserved is the linker region of macro-H2A among species? And is there any disease causing mutation in this region in humans?
Okay, so you see this person down here on the picture? These are very good questions. We already have written them down, and he's in charge, Ollie Mers, to give us the answer in 6 months.
Perfect.
And I can answer the conservation question. We already looked at the conservation. It's not the sequence which is completely conserved, but the length and the positioning of lysins, but not most of the sequence is conserved.
Thanks. Now have a peak of questions that I'm trying to go through. There is one here. Yeah. Regarding CRX, the shRNA inhibitor experiments, what happens if you keep the cells long enough in culture? Do you see resistance, as happens with ACE alone?
We haven't done, the short answer is we haven't done these experiments yet. I would predict that most likely if you do a resistant experiment, until now we have been able to grow resistant cells whenever we tried to push the system. But we haven't done these experiments yet. That's a very good question.
Another one here. Expression itself of macro H2A is shown to be deregulated in leukemia lymphoma.
I mean, if you roughly look at this again, René would be able to answer that question much better. Yes, we see more macro H2A in leukemia cells than in hematopoietic stem cells, but we have not really looked into detail which isoform it is, because most of the data sets we have analyzed do not allow us to discriminate the different isoforms. So we need to do this ourselves.